Dear viewers, this video is part 2 in the 6-part series on the cell and its functions in Medicine for Common Man series. Cell membrane proteins. There are globular masses floating in the lipid bilayer. These membrane proteins are mainly glycoproteins. There are two types of cell membrane proteins: integral proteins, which protrude all the way through the membrane, and peripheral proteins, which are attached only to one surface of the membrane and do not penetrate all the way through. Many of the integral proteins provide structural channels or pores through which water molecules and water-soluble substances, especially ions, can diffuse between extracellular and intracellular fluids. These protein channels also have selective properties that allow preferential diffusion of some substances over others. Other integral proteins act as carrier proteins for transporting substances that otherwise could not penetrate the lipid bilayer. Sometimes these carrier proteins even transport substances in the direction opposite to their electrochemical gradients for diffusion, which is called active transport. Still others act as enzymes. Integral membrane proteins can also serve as receptors for water-soluble chemicals such as peptide hormones that do not easily penetrate the cell membrane. Interaction of cell membrane receptors with specific ligands that bind to the receptor causes conformational changes in the receptor protein. This process, in turn, enzymatically activates the intracellular part of the protein or induces interactions between the receptor and proteins in the cytoplasm that act as second messengers, relaying the signal from the extracellular part of the receptor to the interior of the cell. In this way, integral proteins spanning the cell membrane provides a means of conveying information about the environment to the cell interior. Peripheral protein molecules are often attached to integral proteins. These peripheral proteins function almost entirely as enzymes or as controllers of transport of substances through cell membrane pores. Dear listeners, by the way, this educational video is made possible by Prescribon, the makers of e-prescription software for digital prescriptions available freely for doctors on the website www.prescribon.com. Doctors who are interested in this e-prescription can freely avail of its vast collection of more than 100,000 e-prescriptions. Membrane carbohydrates. Membrane carbohydrates occur almost invariably in combination with proteins or lipids in the form of glycoproteins or glycolipids. In fact, most of the integral proteins are glycoproteins and about 1/10 of the membrane lipid molecules are glycolipids. The glyco portions of these molecules almost invariably protrude to the outside of the cell, dangling outward from the cell surface. Many other carbohydrate compounds called proteoglycans, which are mainly carbohydrates bound to small protein cores, are loosely attached to the outer surface of the cell. Thus, the entire outside surface of the cell often has a loose carbohydrate coat called the glycocalyx. The carbohydrate moieties attached to the outer surface of the cell have several important functions. One, many of them have a negative electrical charge, which gives most cells an overall negative surface charge that repels other negatively charged objects. The glycocalyx of some cells attaches to the glycocalyx of other cells, thus attaching cells to one another. 3. Many of the carbohydrates act as receptors for binding hormones such as insulin. When bound, this combination activates attached internal proteins that in turn activate a cascade of intracellular enzymes. 4. Some carbohydrate moieties enter into immune reaction. Dear viewers, your kind attention is drawn to the figure 1.3. This figure 1.3 shows the cell structures under the greater magnification of electron microscope. The cell membrane encloses nucleus, nucleolus, chromatin, endoplasmic reticulum, Golgi apparatus, ribosomes, and other cell structures. Cellular organelles and the cytoskeleton. The cell is composed of discrete organelles that subserve distinct functions. When a eukaryotic cell is viewed through a light microscope, a handful of recognizable intracellular structures can be discerned. The intracellular matrix or cytoplasm appears grainy, suggesting the presence of components that are too small to be discriminated by this technique. With the much higher magnifications available with an electron microscope, the graininess gives way to clarity that reveals the cell interior to be remarkably complex. Even the simplest nucleated animal cell possesses a wide variety of intricate structures with specific shapes and sizes. These structures are the membrane enclosed organelles, the functional building blocks of cells. Figure 3.1.3 illustrates the interior of a typical cell, as you have already seen. The largest organelle in this picture is the nucleus, which houses the cell's complement of genetic information. This structure, which is visible in the light microscope, is usually round or oblong, although in some cells it displays a complex lobulated shape. Depending on the cell type, the nucleus can range in diameter from 2 to 20 micrometers. With some exceptions, including skeletal muscle and certain specialized cells of the immune system, each animal cell, including the humans, has a single nucleus. Cytoplasm and its organelles. The cytoplasm is filled with minute and large dispersed particles and organelles. The jelly-like fluid portion of the cytoplasm, in which particles are dispersed, is called cytosol and contains mainly dissolved proteins, electrolytes, and glucose. Dispersed in the cytoplasm are neutral fat globules, glycogen granules, ribosomes, and secretory vesicles, and 5 especially important organelles: the endoplasmic reticulum, Golgi apparatus, mitochondria, lysosomes, and peroxisomes. Endoplasmic reticulum. Surrounding the nucleus is a web of tubules or saccules known as the endoplasmic reticulum, hereafter depicted as ER. Endoplasmic reticulum is a network of tubular structures called cisternae and flat vesicular structures in the cytoplasm. This organelle can exist in either of two forms, rough or smooth. The surfaces of the rough ER tubules are studded with ribosomes, the major sites of protein synthesis. Ribosomes can also exist free in the cytosol. The surfaces of the smooth ER, which participates in lipid synthesis, are not similarly endowed. The ER also serves as a major reservoir for calcium ions. The ER membrane is equipped with calcium pump that uses the energy released through ATP hydrolysis to drive the transport of calcium ions from the cytoplasm into the ER lumen. This calcium ion plays a major role in intracellular signaling. This organelle helps process molecules made by the cell and transports them to their specific destinations inside or outside the cell. The tubules and vesicles interconnect. Also, their walls are constructed of lipid bilayer membranes that contain large amounts of proteins similar to the cell membrane. The total surface area of this structure in some cells, example liver cells, can be as much as 30 to 40 times the cell membrane area. The detailed structure of a small portion of endoplasmic reticulum is shown in figure 1.3. The space inside the tubules and vesicles is filled with endoplasmic matrix, a watery medium that is different from fluid in the cytosol outside the endoplasmic reticulum. Electron micrographs show that the space inside the endoplasmic reticulum is connected with the space between the two membrane surfaces of the nuclear membrane. Substances formed in some parts of the cell enter the space of the endoplasmic reticulum and are then directed to other parts of the cell. Also, the vast surface area of this reticulum and the multiple enzyme systems attached to its membrane provides the mechanisms for a major share of the cell's metabolic functions. Ribosomes and the rough or granular endoplasmic reticulum. Attached to the outer surfaces of many parts of the endoplasmic reticulum are large numbers of minute granular particles called ribosomes. Where these particles are present, the reticulum is called the rough or granular endoplasmic reticulum. The ribosomes are composed of a mixture of RNA and proteins. They function to synthesize new protein molecules in the cell, as discussed later. Smooth or agranular endoplasmic reticulum. Part of the endoplasmic reticulum has no attached ribosomes. This part is called the smooth or agranular endoplasmic reticulum. The smooth reticulum functions for the synthesis of lipid substances and for other processes of the cells promoted by intra-reticular enzymes. Golgi apparatus. Kindly see the figure 1.3 for details. The Golgi complex resembles a stack of pancakes. Each pancake in the stack represents a discrete flat saccule. The number and the size of the saccules in the Golgi stack vary among cell types. The Golgi apparatus is closely related to the endoplasmic reticulum. It has membranes similar to those of the smooth endoplasmic reticulum. The Golgi apparatus is usually composed of 4 or more stacked layers of thin, flat, enclosed vesicles lying near one side of the nucleus. This apparatus is prominent in secretory cells, where it is located on the side of the cell from which secretory substances are extruded. The Golgi apparatus functions in association with the endoplasmic reticulum. Small transport vesicles, also called endoplasmic reticulum vesicles, that is ER vesicles, continually pinch off from the endoplasmic reticulum and shortly thereafter fuse with the Golgi apparatus. In this way, substances entrapped in ER vesicles are transported from the endoplasmic reticulum to the Golgi apparatus. The transported substances are then processed in the Golgi apparatus to form lysosomes, secretory vesicles, and other cytoplasmic components. The Golgi complex is a processing station that participates in protein maturation and targets newly synthesized proteins to their appropriate subcellular destinations. And so, dear viewers, this is the end of part 2 in the 6-part series on the cell and its functions in Medicine for Common Man series. Thank you for watching the video.